Prendi parte all'agricoltura del futuro con Agrialghe, i biostimolanti di nuova generazione basati su una selezione ottimizzata di diverse specie di microalghe, combinate nella giusta proporzione che superano le soluzioni esistenti, aumentando le rese, le qualità e la resistenza delle vostre colture. Prendi parte alla biorivoluzione.
Good morning, everyone. We can start this forum dedicated to biosolutions for the agricultural sector. And this year's title is What is New About the Fight Against BMSB. My name is Davide Bernabé, and I am the Biosolutions Development Manager. I would like to thank Macfrut for including this forum in this special digital edition of Macfrut. It's a brand new method to hold such conferences, and I'm thankful for that. Our aim is to offer innovative solutions for biological control, and we want to do that thanks to the experiences of some entrepreneurs and technicians that can share their tangible and first-hand experience with us. Today's guests are going to speak about biocontrol for BMSB. It's a complex topic that we have been facing for several years now, not just in Italy, but in many countries in the world. I would like to thank our guests for being here today, and I would also like to thank our US guests. I'm really grateful to them because it's night now in the United States, and so thank you for being here. I'm going to introduce our guests. We're going to hear their speeches, and then we're going to have a brief Q&A time. For those who are following online, you can send questions by WhatsApp to the following number: 338 36 59 930. 338 36 59 930. At the end of every presentation, I'm going to ask each speaker a question, and I will try to take your questions into consideration as well. I would like to ask our guests to stick to the allotted time, and we will start with Mauro Gavioli, an agricultural entrepreneur based in the province of Modena. He is going to tell us about pear growing and the difficulties that he has experienced in the worst years of the diffusion of BMSB. Mauro, the floor is yours. Thank you. Good morning, everyone. I would like to thank Macfrut for this opportunity. I'm really glad to be here today. I think that it is important to share our experiences and our first-hand experiences with you. It's a very complex issue to tackle. There are different conditions and limitations, and we have no scientific evidence as for BMSB in some fields now. I work in the province of Modena, and 22 years ago I became an organic farmer. I learned by doing, and I had to face many problems and invest a lot of money and time. So I would call myself a pioneer. I think that it is important to bear in mind that there are no ready-made solutions to win the fight against pests. We have to have a comprehensive approach, and we need to invest on prevention as for insects and fungi. in particular. While I was driving here, I was wondering, what are biosolutions? Well, these are techniques that I use on a daily basis nowadays. My core business is organic farming. That is why I use these methods basically every day. What are biosolutions? The release of parasites, the use of nets in orchards in particular, and mechanical solutions that are quite complex. It's not just a matter of spraying. There are even more complex treatments that can be carried out, and later on I'm going to explain to you some strategies that we have implemented. My company has always been focused on research. I think that if we do not find the right solution to this problem, it will be a really serious problem, even more serious in the future. Stink bugs are not the only problem that we are facing at our company, because there is a wide range of pests. And this is a serious problem because there are other fungi and other pests that cause damage to the crops. And this is something not to be underestimated. We can't afford to make mistakes in the agricultural sector. That is why I have been collaborating for a few years with some technicians, and this really made the difference for us. We could share experiences and exchange our ideas. And we also collaborated with some laboratories. Having access to lab tests is very important because this speeds the whole process up, and you can get to the root causes of the problems that you are experiencing. I would like to thank Dr. Luca Casoli, the director of the Phytosanitary Consortium of Modena and Reggio Emilia, because he was of great help in the fight against BMSP. And I would like to thank Mr. Bucchi Renzo, that was able to support us at a very difficult time. We were not able to find a solution, and he was of great help for us. I'm not saying that I have found all solutions, because in 2019 we experienced huge damage because of BMSP, and that affected up to 70% of our yield. Nonetheless, we responded and reacted to that problem, and we tried to understand this problem better in order to perform better in 2020. That is why we were not completely defeated. And this year the damage we had was between 0 and 5%. One more thing. Sometimes I wonder if this year we had the same amount of pests as in the previous year, I think that we would not have achieved such a great result this year, because the pests overwintering in our company this year were fewer if compared to last year. So the strategies we implemented were based on 3 elements: adult pets, migration, and nymphs. We know that adult pests are the most difficult one to defeat because they are really strong and resistant. That is why we implemented some strategies using essences, powders, and enemy fungi. As for migration, we implemented the so-called attract and kill strategy. This means that we worked in order to turn disadvantages into advantages. So there were no woods around our crops, so we installed wooden poles with pheromones on the whole border of our growing area. And we installed some traps both on the poles and on the border bushes. And between May and June, we captured between 2 and 3 stink bugs a day. And so you can understand that the total amount was quite high. As for nymphs, instead, we performed treatments with pesticides, but you cannot use too many of these products. My time is almost over, so I will try to be very brief. It's difficult to find the right balance of insecticides because there are no ready-made solutions to find this balance. You have to use both chemicals and biosolutions, otherwise you will always have a certain damage share, and this will affect also the quality of the product. Thank you very much for your attention. Thank you very much. We have a question for Mr. Mauro Gavioli. First of all, thank you for sharing your experience because you shared some very interesting details with us. I think that there were some hints that you got that led to the use of new treatments. So, if you had to give advice to other farmers, where would you suggest that they start from? Well, first of all, I would suggest to seek for professional advice from the very beginning. You need to cooperate with a team of technicians that can have access to laboratories and the necessary techniques. And this also allows you to save time, because time is vital. So my advice would be to collaborate with professionals in order to save time, because that's the core element. Thank you very much. We will now leave the floor to Mr. Fabio Zannoni, a technical agronomist of Cab Massari, located in Ravenna. He's going to speak about crop protection in his area. Thank you very much. You have the floor.
Good morning, everyone. I'm Fabio Zannoni and I'm a technician for the Cab Massari company in Conselice, Ravenna, in the province of Ravenna. We have 35 hectares of organic pear crops, of which many of which are of William Abate Conference pears and Kaiser pears. We've been producing organic pears for 15 years now. At first, we had many hurdles to overcome, because when we started our organic growing, we didn't have actually any guidelines, any technical advice to follow. So we tried to understand our company's needs, and as my colleague was saying, we tried to share our opinions and views with our colleagues, because only by establishing some networks with our colleagues, you can share your experiences and also get new insights on how to solve these problems. 80% of our pear crops are covered with a single row nets. And it's been 15 years now that we have been using these nets, and they are quite efficient in fighting against BMSB. However, we need to implement some strategies in order to make these nets even more efficient, and that's what I would like to talk about now. First of all, I think that immediately after the fruit set, it's important to close these nets, because in this way we can protect our crops from the adults that are the strongest species, as we were saying earlier on. So in this way we protect our crops. But we also need to pay attention, because using nets can have thinning effects on our crops. So we need to be sure when to close these nets. Secondly, if these nets are installed on plastic caps, we have to pay attention, because these plastic caps can be overwintering places. And so in spring, when we seal the pear with these nets, if the adults are inside our plastic cups, then the crop is completely infested by the BMSB. That's why we tried to close these cups and all the holes that we had in our nets in order to prevent the overwintering. And this was a good solution that we adopted. A third aspect that I want to talk about is related to the importance of avoiding the sprouts from going out the net, because we've seen that the adults lay their eggs on the lids, the eggs hatch, and therefore the nymphs reach the crops, because unfortunately the mesh of the nets aren't so close. And that's the reason why in June we should try to intervene manually and eliminate all the sprouts that were protruding out of our nets. And we reached such a solution last year when we saw that our plants, our crops, were affected the most at the top of their stems, So we asked why, because nets were covering the plants, so the BMSB should have affected them from the ground, but that wasn't happening. So we started focusing on the inlet points, holes that the nets had. So by taking actions and closing all the holes that were in the nets, we have managed to make them even more efficient. So these small strategies that I've just mentioned to you can be very useful, and you can all use them in order to increase the efficiency of these nets that we can use to reduce BMSB attacks. And as my colleague was saying, when we decide to have to grow organic farming, you always have to try to prevent many different problems. And that's why you need to perfectly know the kind of soil that you have and all the features of the crops that you want to grow. In addition, it's important to establish networks, to go and visit other companies, because only by taking these kinds of actions you can improve your crops and you can get more efficient solutions for growing organic products. Having organic farming for pears, for example, can be an added value, that's true. But if you do not have any experience at all, you can suffer from huge damage, because there might be many problems to overcome, especially when dealing with organic farming. And this is due to the fact that minor diseases can impact very negatively all the crops. And this is also due to the fact that when you find a solution to fight against one problem, such as the BMSB, then another problem may arise. So we need to be ready to deal with many problems. Thank you, Fabio. You have a great wealth of knowledge and experience. So I would like to ask you about the trends of the BMSB attacks in your crops this year. This year we had a limited number of BMSB attacks. I think that this was due to our bad weather conditions, because we had a frost period. We have a lot of hangars in our company that we use to storage wheat, and we had high pressure in our crops, whereas this year the pressure was lower. And so I think that having such a lower pressure and lower temperature helped reduce the attack of BMSB. We have different areas grown by pears, and we've seen that the small woods that were around our pear crops helped the temperature to be more mitigated. And so the pressure wasn't only on the pear crops, but also on all the other crops that we have. And so BMSB attacks involved also the other crops around the pear crops, and this was helpful for us. And so the damage was much lower, less minus 5 to 10% than previous years. If we consider other variety of pears, such as, for example, William pears, where we didn't use any nets, we had a better situation. Thank you so much, Mr. Zannoni. We would like now to give the floor to Dr. Helmer, who will talk about biocontrol and biosolutions against the BMSB in the United States. We thank him for being here with us, especially since it's late night in the US right now, and we are more than happy to listen to his speech now.
At my laboratory in Delaware, we concentrate on classical biological control programs for invasive species. And this has included, in recent years, brown marmorated stink bug, also spotted wing Drosophila, spotted lanternfly, and so on. In past years, since the introduction of brown marmorated stink bug to the U.S., we have conducted considerable research on biological control, trying to find a solution for this stink bug, biological solution, and this research has been a very collaborative effort with many researchers in different organizations, state departments of agriculture, universities, and other government agencies. And a lot of funding has been made available for our biocontrol effort in the U.S. Funding provided by various grant programs from USDA, annual farm bill, producer and grower groups, and many graduate research projects have focused on brown marmorated stink bug. We know from our research in Asia that the primary natural enemy, the most important natural enemy of brown marmorated stink bug in the native range of Asia is the parasitic wasp, Trisulcus japonicus. And much of our research during the first years in North America concerned evaluating the potential safety for release of Trisulcus japonicus in North America against stink bug. While we were conducting this evaluation research, however, we discovered that Trisulcus japonicus had found its way on its own to North America and established in several different locations. We know from our genetic studies that there are at least 3 different introductions of Trisulcus japonicus in different parts of the USA and now in Canada. This chart shows the location of states where Trisulcus japonicus has established or has been found in North America and in Canada. So we know that these adventive or self-introduced populations of Trisulcus japonicus are establishing in a wide range across the U.S. We know also that this is still a process in the early stages. Many of our research activities have shifted now toward promoting naturally occurring populations of Trisulcus japonicus and increasing their effort. So in many of the states where you see here that Trisulcus japonicus is established, efforts are in progress to increase their local abundance, their spread to neighboring states, their spread into agricultural areas, and to try to increase their biological control impact. In each state, the effort is managed a bit differently. Sometimes they're university researchers, sometimes state departments of agriculture are involved. We know that in the U.S., the local population densities are still quite variable across the areas of establishment. So in some areas, Trisulcus japonicus seems to be becoming quite abundant, and in other areas, populations are still small and spotty. Now, in Italy, similar situation is in progress. We know from surveys undertaken by Italian researchers that Trisulcus japonicus has found its way to Italy on its own. In addition to Trisulcus japonicus, a second species of Trisulcus, Trisulcus mitsukurii, has also become established. And these appear to be establishing broadly in Italy. And there are similar redistribution efforts currently underway in Italy to increase their spread, their their local density, abundance, and their impact on brown marmorated stink bug. And this, of course, will be a subject of the following talks, so I won't say more about that here. Of course, with classical biological control, our primary goal is to establish a well-adapted parasite in the environment so that we can rely on the parasite to find its way to populations of pests that we wanted to control. This chart shows some of our options for working with biocontrol agents. This chart comes from a publication that's in press right now by Againstthe and co-authors on reviewing the biological control possibilities for invasive stink bugs. In addition to purely classical and innovative biocontrol programs, we have options now, various options for conserving populations of existing natural enemies, in addition to improving the activity of introduced biological control agents. Now, Trisulcus is a large genus of parasites with many different species, all of which seem to be specialized on stink bugs. And in North America and in Europe, there are native species of Trisulcus and other genera of parasites that do attack brown marmorated stink bug to some degree, although they're not as effective as Trisulcus japonicus is in Asia. There are opportunities for conserving and improving populations of these naturally occurring agents, in addition to relying on semiochemicals produced by the pest stink bug and by plants that the stink bug feeds on to improve the populations of both the introduced Asian parasites and the native parasites. Other approaches include manipulating populations by encouraging the survival of the adult parasites with oral resources, for example. And then, of course, there are a number of opportunities for integrating the biocontrol activities with other behavioral methods for control of stink bug, methods which manipulate the population of stink bugs by manipulating their behavior. Tracy Lesky and other speakers will talk more about this, I'm sure. I'll focus a little bit on the semiochemical aspect of biological control. And of course, semiochemicals are widely used in brown marmorated stink bug management programs as tools for monitoring their population by using pheromones and lures. And we know, fortunately, that the lures that are used to monitor BMSP populations are not attractive to Trisulcus parasites. Good thing, because we don't want to be pulling these parasites out of the population, out of the environment into the lure. So the traditional methods that are being used now to monitor these pyramid traps, pheromone traps, clear sticky traps with pheromone lures attached, are good methods for monitoring without impacting the population of natural enemies. But I do want to mention a caution that parasitic Hymenoptera, including Trisulcus parasites, are highly attracted to yellow sticky traps. So it's important to realize that in situations where yellow sticky traps are being used for monitoring, that these can have an impact on the local parasite population. So they need to be used cautiously if Trisulcus are being used to augment natural enemy populations. Now, there's a large body of research that shows that stink bug feeding on plants and even their oviposition, the deposition of egg masses on the surface of leaves, causes plants to emit a range of volatile organic compounds, VOCs. A lot of this research has been done by Italian research groups in recent years. We also know that as stink bugs, including brown marmorated stink bug, walk on leaf surfaces, their bodies are leaving chemical traces on the leaf surfaces, which can persist for some time, even for several days. There's a large body of entomological research that shows that many species of parasites can detect volatile organic compounds that plants emit when they're being fed on by insects, and the parasites can often detect these volatile organic compounds from some distance, and they can follow these as cues to locate plants that have their host insects on them. Once the parasites find their way to these plants, when they're searching on leaf surfaces, they're also able to detect the traces that the BMSB and other insects leave behind when they're walking on the plants. And these traces serve as cues to the parasites that their host insect, and in the case of Trusulcus japonicus, probably their eggs are likely to be nearby. These cues alter the behavior of the parasites in ways that increase the probability of their finding these host insects. At our laboratory, we've been studying the influence of these pheromones on the foraging by Trusulcus japonicus. In the laboratory, studies use video cameras to follow the movements of female Trusulcus japonicus on leaves in experimental arenas. And in this example of research shown by one of the graduate students, Sean Boyle, he exposed Trusulcus japonicus females in these small arenas with pieces of leaf that had been treated with adult brown marmorated stink bug and another native stink bug, Podesis maculoventris, which is an important predatory species. It's a species that we do not want Trusulcus japonicus to attack and have an impact on. And he treated 3 different kinds of leaves with pheromone by allowing the stink bugs to walk over the surfaces, and then he observed the behavior of the wasp using the video camera. And what he found showed that Trusulcus japonicus females readily detect the presence of these cues on the leaves and causing them to change their searching behavior to spend more time on leaves that have the traces. And the more time they spend searching, the greater the chance that they will find egg masses that the stink bug has left behind on those leaves. He also found that Trusulcus japonicus could detect the presence of pheromones left by the predatory stink bug, Podesis, but it did not respond as strongly to those pheromones. We believe that this is one facet of specialization which allows Trusulcus japonicus to focus on brown marmorated stink bug in the field and not focus on other species of stink bugs. This work was followed up, led by a graduate student, Robert Malek, graduate student from Italy, and subsequent work in the laboratory identified the chemical components of these pheromones that were left behind by walking BMSB females that are detected by Trusulcus. And the interesting finding is that there are 2 primary compounds, E2-decenol and triticane, that seem to be the components that the wasp is detecting left behind by the stink bugs. You can see from this chemical ID trace there are small peaks of other compounds that are not yet identified, but the 2 primary components are the 2-decenol and the triticane. These are interesting because they are also produced by other species of stink bugs and indeed other types of insects. So they seem to be primary cues that natural enemies are using to detect and recognize the traces of their hosts. But the specialization depends on the ratio of the 2 chemicals and the presence of perhaps these secondary compounds in smaller amounts. And the different combinations of these chemicals allow a natural enemy to determine the species of host, the species of traits that they're detecting. Next, we wanted to take these studies into the field to see how the lab studies translate into actual field situations. We first began looking at the ability of Trissolcus japonicus to disperse in the field, and we did this by setting up grids of release experiments, this shown by this example. Here we have 49 potted trees laid out in a star grid system on lines at intervals out from a central point. And we released adult Trissolcus japonicus at the center of the circular test pattern and measured their ability to find egg masses, sentinel egg masses, which we attached to the leaves of these potted plants. And then we discovered that very typically these wasps rapidly disperse out. When there are plants nearby, they will land on them. Within 24 hours, nearly all of the wasps released dispersed from the release experiment into the surrounding environment. So they move quite readily, and they're capable of finding the egg masses on the plants as they move out. We next wanted to determine, could we show whether or not contact pheromone on plants in the field influence the finding ability or the success in finding egg masses. So in this follow-up set of experiments, we placed brown marmorated stink bug in screen cages on half of the 49 potted plants inside these sleeves for 24 hours. We allowed the bugs to walk around. Then we removed the cage, we removed the bugs, and we released wasps in the center of the experimental plot. This chart shows just one example of a result, just one of our replicates. And you can imagine that in any release on any given day, the wasps are highly sensitive to the local weather conditions. So they're very much more active on a warm or a hot sunny day than they would be on a cool cloudy day. And if it's windy, their dispersal is going to be highly subject to the direction of the wind. This plot, you can see that there was a bit of wind from the northeast, so that the red dots, which indicate the sentinel egg masses that were discovered and parasitized in this particular replicate, were influenced by the direction of that wind. Now, so far, these experiments are still in progress, but the results are encouraging. On average, we see that a significant proportion of egg masses are parasitized by this single release of parasites within a 24-hour period. Most of the females that are released into the center of their plot disperse outward into the surrounding area, and only a few of the females that are being released are responsible for finding the egg masses on the plants in this center plot. But nevertheless, it's resulting in significant parasitism. And we do see that the plants that have the pheromone on them from the stink bug are being found at a significantly higher rate than the egg masses that did not have the stink bug traces associated with them on their plant. So it does appear that the pheromone is an important cue, and it operates under field conditions to improve the ability of Trissolcus to find their hosts. This raises the possibility that this pheromone can be manipulated for potential field use, and we're currently looking at how they respond to application of actual E2-decenol and tridecane solutions applied in the field to see whether we can manipulate and increase the parasitism of stink bug. And we're also continuing to investigate the response of Trissolcus to non-target stink bugs, such as the predatory podesis, under field conditions. So in North America, there's a lot of research, a lot of interest in this subject, and it's not at the point where we can use it in the field under commercial conditions, but we're certainly moving in that direction with our research. There's also larger scale research trying to introduce Trissolcus japonicus into commercial scale situations. And there are area-wide programs, such as indicated in this example from Tracy Lesky's lab, in which Trissolcus is introduced near commercial crops. These studies are also in the early stages, but the results do show that the Trissolcus are capable of finding and parasitizing stink bug egg masses, in that even in situations where these egg masses may have been treated, sometimes the egg masses will produce Trissolcus japonicus even though the eggs have been sprayed, although survivorship is clearly highest in unsprayed refugia, areas around the orchards, for example, that have natural vegetation, or in border areas that may not be sprayed. So Tracy perhaps will talk a little bit more about some of this. One final area that I wanted to spend a minute or two talking about is the possibility of utilizing flowering plants to improve the longevity of Trissolcus populations that are in the field. And again, there's a large body of scientific research that shows that parasitic Hymenoptera benefit from having presence of flowering resources around crops, because the parasites use these flowering resources to feed on their on the nectar, which provide carbohydrate and reserves and a moisture source, which improves their longevity. And we know that Trissolcus japonicus from lab studies can survive for several months, basically for an entire field season, if they're supplied with moisture and a sugar source, usually honey in lab studies. So in studies at an ARS lab, in Jana Lee's lab in Oregon, she has been investigating the influence of different floral resources to improve the longevity of Trissolcus japonicus populations. And many of the flowering resources that have been investigated in other insect pest systems do not prove to be very important at or useful at improving Trissolcus japonicus longevity, but there are certain species of plants that she did find were very useful, highlighted on this chart. In particular, buckwheat, cilantro, and dillweed. Each of these species improved the longevity of Trissolcus japonicus in some cases up to 2 months under field conditions. So this again raises the possibility of using floral plantings in border areas or as understories for crops to improve the conservation of Trissolcus japonicus populations in the field. But returning in summary, there are many opportunities for using insect semiochemicals and plant semiochemicals, flower resources, and so on to improve the utilization of Trissolcus japonicus and other native Trissolcus. Going forward, this research will be continuing, and we hope that in the near future some of these techniques can be utilized in commercial ecosystems. Thank you very much.
Thank you. Grazie
al dottor Elmer per il dettaglio che ci ha voluto dare di tutte di queste ricerche veramente interessanti che sono state svolte negli Stati Uniti. E gli vorrei chiedere una domanda che ci è giunta tra l'altro dal dottor Gavioli, dal signor Gavioli, che è qui con noi oggi. E se nelle vostre esperienze
by Mr. Gavioli, who is here with us, we would like to know whether, when considering the areas where you discharged the most, where you released the most, these Trissolcus japonicus, if you managed to control the overwintering populations even during the first years after this release. Thank you.
Actually, I think the release program for Trisulcus japonicus in Italy is actually a larger and better organized program than the different releases of Trisulcus japonicus in North America. From what I have heard, the numbers released and the number of sites within a short period of time this past year are very impressive. In North America, the results in each state are still variable. Again, the populations of Trisulcus japonicus in many of the parts of the U.S. where we've been conducting research are still rather low. We do see relatively high levels of attack in several of the areas, but we can't really generalize to predict what the impact in commercial settings will be, because most of these monitorings are in natural habitats still in North America. So we're not yet at the point where we can predict the impact of Trisulcus japonicus on BMSB in the U.S. But we're very hopeful, because in Asia, where Trisulcus japonicus has been collected for a number of years in our surveys for natural enemies, we routinely see egg parasitism by the end of the season of 75 to 90%. So we fully expect that eventually, when Trisulcus japonicus is fully established in the invaded ranges, that we will see similar levels of impact. But we're not at the point where we see that yet.
Ok, Kim Elmer, thank you very much. Grazie mille Thank you very much. Grazie mille al dottor Kim Elmer per questa possibilità.
Thank you again for giving us the opportunity to listen to your very interesting speech. I would like now to give the floor to Mr. Luca Casoli, who is the director of the Phytosanitary Consortium in Modena and Reggio Emilia, and who is the coordinator of the project on the fight against the BMSB by using the Trisulcus japonicus that Mr. Holmer has just talked about. This program is part of a national program that involved many different research activities and also national ministries. So we would like to ask Mr. Casoli to talk about this project, to describe its layout, and we would like to ask him which are the opportunities that he sees for the future. Thank you. Good morning, everyone. I'm Luca Casoli from the Phytosanitary Consortium in Modena and Reggio Emilia. I thank the organizers for inviting me here and also for organizing this very interesting meeting focused on the fight against BMSB. The activities that we have carried out over the last year has been quite successful and has seen the involvement of many bodies, regional institutes, technical institutes, and also universities. 1 year ago, the Emilia-Romagna region decided to take part in this project, to start this project. In order to manage and to handle the fight against BMSB by using Biosolutions. The project was based on the idea that this kind of species was controlled in a indirect way. And so the actions taken were indirect. And these systems were integrated with other actions. So we had both the use of nets and also the use of biological solutions to fight against these pests. It was quite difficult as far as the Italian law was concerned to put into place such a project, but eventually we managed to launch this project, even though there were a few interruptions over the time. And the National Research Institute, as I was saying, eventually managed to launch this project together with the regions involved. We can say that the activities started in 2019 when we identified some structures that could work together, our consortia and our research institute, in order to breed these species. So we worked in close collaboration both with the University of Bologna and with the University of Modena and Reggio Emilia, and also with other regional institutes. And this project was started. So we tried to collect, to find overwintering bodies, overwintering species that could be used at the beginning of this year in order to multiply the presence of Trisolcus japonicus species. So the number of Trisolcus japonicus has tripled, and we also monitored the area involved because we wanted a sort of a situation in which we were able to identify the areas in which the Trisolcus japonicus species were in a higher number. And we also tried to design a new net with mesh at a distance, at a very large distance. And we tried also to identify the areas in the Emilia-Romagna region where we could release the Trisolcus japonicus. And we identified 300 areas that were adapted for this purpose. And our aim was to allow the Trisolcus japonicus to go and live in these areas, green areas that we have identified that were near orchards. And the precise moments that we chose for the release were in June and July. And in these two periods, we have put 100 females, and we didn't put any male species. And we had to stick to a verification protocol in order to assess the impact that these species had on the areas where it was inserted. Then a monitoring action took place. So we carried out survey to see what happened 20 days after the first release and then 20 days after the second release. And we also carried out detailed analysis and surveys by our universities in order to to calculate the number of eggs that could be found after the release. We've just finished calculating the number of eggs, and we will be analyzing all the results over the next few weeks, and we hope that we can carry out a very detailed analysis about the eggs. We are also carrying out an analysis as for the agricultural aspect of our soil in order to check and assess the environmental resilience of our environment. We were talking about the experience in the United States earlier on, and we want to follow their example and provide detailed data about the monitoring of the situation here in Italy. We are happy because this ambitious project has almost reached its end, and so I'm very glad, and I want to thank all the people who were involved in this project. It's quite difficult now to draw some conclusions of this project, but we will go on carrying out analysis and surveys to monitor the evolution of these species and also the reaction of the environment in order to implement a real integrated control program of BMSB, trying to put into place both direct and indirect protection actions, because the final aim is to have fewer BMSB attacks over the following years in our orchards and crops. Thank you. Thank you, Mr. Casoli. I have a question for you. What's your opinion as for the trend of the BMSB population this year? Because we've seen that the population trends over the years have changed a lot. And which are the elements that we can look at in order to predict what will happen in the future? Thank you. We have been monitoring this species very careful since 2012, when it first arrived here in Italy, in order to predict its evolution, of course. And we've seen that the population trends have changed and varied a lot over the years. This was due to different problems, such as weather problems. And in 2017, for example, there was a fall in the number of BMSB here in Italy. And so we were quite positive because we thought that these species had some problems in living here in Italy. But then in 2018, there was a rise in the number of BMSB. And in 2019, we reached the peak of the population of these pests that caused a lot of damage. So this was just to say that there are a lot of variations as for the population density of these species. So it's very difficult to predict the future. In 2019, autumn 2019, there was a slight fall, as I was saying, in the number of BMSB. But then we also have to say that spring was even colder than winter, and we also had a very dry period in May. where rains were absent. So all these weather elements, weather forecasts, maybe had positively impacted the presence of these BMSB that were less numerous in Italy in that year. However, we do not have any tangible data to talk about the results and effects of the results by using biosolutions, so by using the parasitization of some species. So it's very difficult to predict the next year evolution, next year trend. We've seen that over the past years, if we have favorable weather conditions, then the population density could be lower. So we've seen the impact that the weather has on the population density. But then it's very difficult to predict future trends. In the past, when we had lower temperature, the number of these pests increased again. So it's very, very difficult to give you a prediction about the future. Thank you so much, Mr. Casali, both for your speech and also for sharing your experience with us. Because we've seen that it's possible to have some strategies to deal with such pandemics. Because, of course, there will be other pests in the future that will affect our crops. So we need to know how to react, and we need also to stress the importance of networking in order to find solutions. And Professor Casali also highlighted the importance of weather conditions in order to fight against these pests. The collaboration between the private and public sector is also another example through which we can carry out actions to fight against pest-related problems. And the National Research Center implemented valuable actions to fight against the BMSB. I would like now to give the floor to Dr. Tracy Laskey, Director and Research Leader in West Virginia. She's been working a lot in developing and implementing IPM tactics for the invasive brown marmorated sting bug. So I would like to thank her for being with us today. And we would like her to talk us about a little bit about all the strategies that they have implemented to fight against the BMSB.
Buongiorno.
So first of all, hopefully you can hear me and see my slides, but I'd like to say thank you to the organizers for the opportunity to speak to you today about our work with the invasive brown marmorated stink bug and development of IPM tactics for their management. I'm going to just go ahead and go and talk, and hopefully you can see the slides. So again, thank you for joining us today. I'll begin by talking about the 2010 outbreak that occurred here in the US, where we had severe problems from this species beginning as early as 2008 and 2009. But in 2010, severe agricultural problems emerged. And this caused significant problems for our fruit growers. We had growers that lost over 50% of their stone fruit crops, both peaches and nectarines, in 2010 from the season long problems. And for our apple growers, it was equally devastating, over $37 million in injury from this pest to apples in just 5 states. And it was during that year in 2010 when we recognized that this pest was really a landscape level threat, because not only could it be feeding and reproducing in an apple orchard, but also in native woody hosts, in cornfields, and on invasive plants like tree of heaven. Since 2010, brown marmorated stink bug has spread across the United States. In most states in the country have now had detections. However, most of the injury has been concentrated in the eastern United States, with the severe injury in our region, where both Dr. Holmer and I are located in the mid-Atlantic. When this crisis hit, we really had to come up with key questions to sort of frame our research based on short-term and long-term needs. Our short-term needs really had to do with asking, what insecticides can we use to manage brown marmorated stink bug to essentially put out the fire? After that, asking the question, how can we monitor this pest and make management decisions to reduce these insecticide inputs and increase sustainability? But also, then finally, what other IPM tactics can we use? And so that's how I'll frame the rest of this talk. So our early work was really asking, what insecticides could we use? And we found a number of compounds that were very lethal to brown marmorated stink bug. And they really belong to just a couple of classes: the carbamates, the pyrethroids, and the neonicotinoids. And so again, these were quite lethal against brown marmorated stink bug, but there were 2 major drawbacks. One was that all of these had very short residual activity. And the second, of course, is that they are very hard on our natural enemies, with most being harmful, or at least being, we would recommend caution in their use because of their impact. So the question was then, how can we move away from these insecticides? And this really gets into monitoring or biosurveillance systems using traps, and asking the question, how can we monitor brown marmorated stink bug and make pest management decisions that reduce insecticide inputs and increase sustainability? And so there really are 4 key components that we looked at. They included a visual cue, which is that black trunk mimicking stimulus provided by those pyramid traps, followed by an olfactory cue, which was based on the identification of the pheromone and pheromone synergist that we were able to then formulate into lures to use in these traps. Third was a capture mechanism based on the insect's behavior, in this case, a jar top with a killing agent to retain trapped bugs. And then finally, a deployment strategy. And we found by using these traps, you could deploy them on the border or interior of orchards and still to really understand and to be able to determine their presence, relative abundance, and seasonal activity. So that was good, but the question was next, can we use these baited traps to guide our management decisions for brown marmorated stink bug? And so some of our experimentation with these pyramid traps, we deployed them in the center and exterior of plots. And what we did was to set thresholds for insecticides to be applied when, excuse me, adults in traps reached 1, 10, or 20 adults per trap. So when we captured that many stink bugs, we would apply an insecticide. We compared this with blocks that we sprayed weekly and a no-spray control. And what we found was that, not surprisingly, the higher the threshold, the fewer the sprays that went on. But the question was, would this also this reduction in sprays also protect fruit? And so what we found was that sprays applied when traps reached 10 adults per trap reduced sprays by 40%, but protected the fruit as well as sprays being applied weekly, which was really encouraging, and it told us that we could use these traps to guide our management for brown marmorated stink bug in apple orchards. Since that time, we have transitioned to clear sticky panels deployed atop wooden posts. These still provide that visual stimulus, but they are cheaper and easier to deploy, and we've been working on a threshold for their use in apple orchards. To date, we have been able to show that a threshold for these sticky panel traps is about 4 adults per trap, cumulative. They've reduced sprays by about 50% in our experimental trials, and working in commercial orchards, they've also worked well. And so if you can see this slide, this particular grower had about 25 stink bugs on their trap, and we would tell them to spray immediately. We also went on to look at other IPM tactics and their development. So I've been talking about this trap-based treatment threshold, where when the trap reaches a threshold, we spray the entire block. And certainly this does reduce insecticide inputs and labor, but we wanted to reduce this further. So we have worked on attract and kill and border sprays. In the case of attract and kill, we baited select border row apple trees with the pheromone, and then the grower would treat these with an insecticide on a weekly basis. Certainly we reduced the insecticide inputs, but in terms of labor, the weekly sprays were really difficult for growers to keep up with. It worked well, it reduced sprays by about 70%, but too much work. We've run into the same issue with border sprays, where in this case, the border of the orchard is treated weekly, and this also can work well. It reduces insecticide inputs, but again, that weekly labor is difficult. And in the case of these, we also back these up with traps in the center to determine if these fail, should we need a full block insecticide application. But again, the key thing that we've been concerned about over the last few years in trying to make adjustments is to deal with this labor issue. And so we've been transitioning from, at least in the case of attract and kill, from spraying baited apple trees with an insecticide to these long-lasting insecticide-treated nets, which also have been used in Italy. And so our work has been really trying to determine where these nets should be positioned relative to the orchard to maximize their impact against BMSB based on brown marmorated stink bugs behavior. And so we put these in 3 locations and evaluated them over the past 4 years. We have placed these long-lasting insecticide-treated nets outside the orchard, about 5 meters away, next to orchard trees right at the border, and we've hung them on trees themselves. In terms of our results, we've found that nets deployed outside the orchard are less effective, with injury rates generally higher than sprayed blocks, sort of generally sprayed orchard blocks. And this really is because we have decoupled the long-lasting insecticide-treated net from the host plant, which is the apple tree, and the pheromone. They really all need to be closer together to maximize the attraction, retention, and kill of brown marmorated stink bug. So what we found is either by deploying them next to the tree or right on the tree, we can see that this is much more effective, that injury is lower to or equivalent to sprayed orchard blocks. And so again, it has to do with maximizing the attraction, retention, and kill of this system. So in terms of our next steps, we still need to remain vigilant. Even after 10 years, we still have damaging populations in the mid-Atlantic where I work. And we're seeing this continue to be a problem. It's not like 2010, but certainly growers are still treating for this pest. We're continuing to refine our IPM tactics and promote grower adoption. About 3/4 or 75% of our growers are interested in attract and kill and border sprays and using that threshold-based trapping system. But ultimately, we all know that we need to integrate long-term landscape level biological control tactics, as Kim and other speakers have mentioned. With that, I'll just say thank you and sorry for the technical problems, and I appreciate the invitation.
For your very interesting speech and for all the tangible experiences that you've shown us. We would like to ask you how you organized your activities in order to give the farmers all these protection strategies. So how did the farmers and farms react to all the strategies that you have detected, and how did you help them to get to know your research results?
Sure. So initially, in order to begin to promote adoption, when pheromone products were really very much experimental, they were still being made, for example, by our research chemists. We would do trials on their farms and work with them collaboratively to allow them to see how it works and gain feedback from them to determine how best to then sort of modify the system. We also have an extensive what's referred to as our extension collaborators. So these are researchers who have specific responsibilities to assist with adoption and promotion of IPM tactics. And so these collaborators also help promote adoption to growers by demonstrating on farms how these products work. I can tell you that we are still we still have a long way to go in terms of adoption. Part of this has to do with the fact that commercially available products that are really, we'll say, fine-tuned and we know how they work, have only been available for about 2 years or so. And so I expect over the next few years, we'll see grower adoption, especially in areas where brown marmorated stink bug continues to be a problem. In surveys that we've conducted, growers indicate anywhere from 66 to 75% willingness to adopt IPM tactics on their farms to reduce insecticide inputs.
Thank you very much. Grazie mille alla dottoressa Leschi.
Thank you very much. We will now leave the floor to Ms. Elisa Marchetti, entomologist. She has been working for many years for the biocontrol of pests, and she currently works for Againstthe 2000. We would like to hear the results of her studies about these pests.
Good morning, everyone. I would like to talk to you about some of the tests that we have been carried out since 2017 at AgriNet. We have carried out some biotests and with different final purposes, as you can see from this slide. Here in this slide you see the main features of Haliomorpha hylis, even though you already know them very well. But I would like to underline why it is so dangerous. At first, we tried to control the adult number, but despite using active principles, we didn't manage to obtain successful results. And there are many factors for these. First of all, it's very particular, specific features and ethology, and it's also very difficult to predict its movements. So all the tools that we have available weren't so successful and so efficient. I would like now to show you the different areas on which I work: the control of nymphs and eggs, the reduction of symbiotic bacteria that are on the surface of the eggs and that are necessary for the production of nutrients, without which the bugs have difficulty in developing or risk dying. And then we also focused on the introduction of the parasitoids, such as, for example, Trisolcus japonicus, here in the Emilia-Romagna region. But today I will focus more on the control of nymphs and eggs. I would like now to show you the results of some of the tests that we have carried out. The first test we did involved the use of Boveriana bassiana. The strain was ATCC 74 or 40, and the results were quite interesting because in eggs treated with this Boveria bassiana, there was a total mortality of around 75% compared to 20% of untreated eggs. And the activity of the product was also detected on newly shelled nymphs. And so the total mortality was around 90%. This test took place in ideal conditions for the development of these fungus. And we need to repeat this test on the fields again in the future. Then we carried out some tests with rock dusts, that is, corroborating agents. We used some nymphs of second age. The product was applied on the nymphs and on their food, and then we carried out controls 2, 4, and 6 days after the application. The results are the following. With the powdered product, there was an overall mortality on day 6 of around 60% versus 4% of the untreated sample. So the mortality rate can be related on the effect that this product has on this insect that becomes drier, as you can see from the images here. Then we carried out some tests with a mixture of pesticides. So we mixed a pesticide based on acetamiprid with some essential oil of orange. Here again the eggs were treated, were sprayed, and we checked the mortality of the nymphs up to 21 days after the application of the product. The total mortality again was about 22%. And we had 54% of ovicidal activity, and this is very interesting, and 31% of activity on nymphs, against a mortality in the sample not treated of 22%. As for the tests carried out with products with antimicrobial activity, we used tripartic copper sulfate together with the essential oil of orange. The methodology was the same as the previous one, so the eggs were treated and we checked the mortality rate. we can say that the combination of copper and orange oil gave very satisfying results, with a total mortality rate of 64% compared to 22% for the untreated sample. Here again we see another test where we used copper and sulfur. The eggs were treated here as well, and here you see all the different doses of the ingredients used. The methodology applied was the same, and we managed to measure and assess the mortality rate also for the adults. And both products used have given satisfactory results, with a total mortality rate of around 60% compared to a mortality rate in the untreated sample of 30%. So we can say that the tests conducted in the laboratory have shown the good efficacy of some products, both on eggs, such as, for example, the Bovaria bassiana and the acetamiprid and the orange oil, and on the nymphs, such as, for example, with the use of rock dust, which will have to be confirmed in the field in the future. Thank you so much for your attention. Your experience and your research has shown us how we can fight against these pests, and the lab activities carried out to fight against these pests. I would like to ask you, Elisa, how long does it take to carry out similar tests? And is it difficult to work with insects inside a lab? Well, it takes longer when we do not have the insects in the laboratory, and having breedings in the lab is not a simple task. So we need to be patient, because there are some problems to overcome, and very often when we want to start breeding some insects, we can rely on previous publications, but other times we have just to test all the different options that we have and see which one is the best one for us. So we need to find out new strategies for the environment in which we are working. And we need to find out the best environment possible to develop and to breed the insect. Once you have the insect, then you have to carry out the test and do the controls. Even with Haliomorphus halis, the main difficulties that we had were a lot, because the mortality rate in the lab is very high. We need to have many different kinds of soils, and so the challenge is tough. Thank you so much. I would like to end this meeting today by leaving the floor to Mr. Vittorio Veronelli, that is member of IBMA Global, and he is a global promoter of the bioprotection in order to protect our crops from pests. So, Mr. Veronelli, could you please tell us what you are doing in order to fight against these pests? Thank you.
Thank you very much. Thank you very much for the invitation. Let's see if we can display my PowerPoint presentation. Thank you very much. Many researchers and technicians have given their speech today. We represent the industry, the manufacturers of biocontrol products, so we are the last ones to speak. The term biocontrol has gained importance over the past few years at an international level in order to standardize techniques and tools globally. Our previous name was Association of Bioprotection Manufacturers, and our current official name is International Biocontrol Manufacturers Association, due to this change at an international level. Our products are natural or plant-based, and they contain macroorganisms such as non-target insects and microorganisms too. And we are adding more and more strategies to our tools. Our association encompasses any technology which is included in national and international regulations and standards. And so our techniques are mainly based on macroorganisms, microorganisms, semiochemicals, and natural substances. We have been working for 25 years. Our company was founded in 1995, and it's made up of new companies, small and medium-sized enterprise mainly by 70%, and there are also many foreign members. In Italy we have 26 companies belonging to the association. The national group of companies can be formed after a certain amount of national members are reached. We are present mainly in Europe, but we also have a global federation called Bioprotection Global, encompassing 7 associations all over the world. Some figures about our position on the market of bioprotection, a field that is gaining more and more importance nowadays. These figures were collected in 2018, and they refer to macro areas in the world. The major market shares are in the US and in Canada, but we also took the period between 2014 and 2018 into account, and you see that there was a constant growth, and we estimate a total amount of 3.5 billion euros a year. This growth has, of course, been supported by policymakers and the decisions taken at a European and international level. The European Union has been implementing many strategies, such as the so-called farm to fork strategy or a zero pollution strategy. Of course, we are speaking of very ambitious strategies that need to be implemented correctly. This pyramid represents the IPM strategy of our association, and as you can see, biological control plays a vital role. However, there are also other systems used in combination with biological control that are also very important. And we are speaking of techniques that have been used for many years now. The Green Deal policy of the last few years have supported this trend, and very ambitious goals were set, for example, minus 50% of pesticides. And many countries are trying to shift towards the achievement of these goals. And this trend is also contributing to updating regulations and standards. We think that we need, of course, standards and regulations. However, we also think that they should be adjusted to the current needs that we have. And time is also a vital factor. 30 years ago, when it comes to biological control, research was time-consuming, so it took very long to find a solution. But now, thanks to that research we carried out in the past, we gained experience and know-hows, and now we can find solutions much quicker. However, these solutions are very often hampered by red tape and the standards set forth in the regulations, and that is the problem. So I think that regulations should set standards in a quicker way, because there is this kind of conflict between the pace of research and the pace of regulation. Biocontrol is the combination of the study of the biological behavior of pests in order to find the weaknesses of pests in order to develop an efficient solution for growers. We all know the advantages of bioprotection. We have a more resilient agriculture. And in terms of costs, I think that we cannot take one single season into account. We need to base our decisions on a longer period, taking the whole system into account. Then there are also the so-called technological convergencies to consider. The term biocontrol was limited because there were new innovative techniques that were developed and brand new IT solutions to apply biocontrol methods or even artificial intelligence. We are speaking of technologies that improve bioprotection means. Just a few words about BMSB. I think that these pests showed us that the environment has a different resilience to the introduction of so-called exotic enemies, depending on the specific situation. And there was a ministry decree approved here in Italy in April 2020. But it is important to speed up the whole process in order to support all the European growers, because sometimes there are different regulations in the different European states. All the companies producing useful tools and technologies in these fields are members of our association. And so I'm sure that they have the necessary know-how, the necessary resources to manufacture efficient tools. However, they must be able to do that with the right timing, and so they need partners to do that, to get to a fast solution to the problems we are facing. Thank you very much for your attention. Thank you very much, Mr. Veronelli. I think that your role is very important and is also the right conclusion, I think, for today's technical forum, in which we have tried to tackle different aspects of these issues. I would like to ask you a question which is not a technical question, actually. You spoke about your strategies to market some solutions. We know that a very important factor in the agricultural sector is the yield of crops. So my question is, what outlooks do you see for the future of organic farming? Because I know that organic farming is an important element of your activity. Despite the decisions of policymakers, biocontrol products are not only used in organic farming, maybe they are even more used outside of the organic farming sector. They are used to a certain extent also in organic farming, but not just in organic farming. There is a huge share of non-organic farming that uses biocontrol products. And I think that this is also the result of the efforts made by the manufacturers of these tools, because they worked really hard to produce sustainable products that are also affordable for companies. We could define them resilient tools that can prove their efficacy over time. I think that there are some very good examples of biocontrol systems that are still proving very efficient, even in the long term. So this is important also for the economic investment of growers and companies. Yes, that's very interesting. Thank you very much. We are getting to the conclusion of this forum. I would like to say that the topics we discussed can be very useful in order to give tangible solutions and advice to fight against BMSP. They were also very useful to give an overview of the different opportunities that are available to continue working in the next few years. And I'm also sure that some of these experiences, such as the one about the national and regional policies that were implemented in order to fight against the very high presence of the BMSP, can show a role model, an efficient model to collaborate between private and public sector to solve this problem. Agri2000net is a partner of Macfrut as for Biosolutions, And this year's edition is focused on this kind of techniques. On macfrutdigital.com you can visit the digital fair trade that has been organized despite the difficulties we are experiencing due to the COVID-19 pandemic. We received many questions on WhatsApp, but unfortunately we did not have the time to answer them. However, we will try to answer them after the end of the conference, and we will try to send the answers. Thank you very much. Again, have a nice day.